Hi everyone, I am Anasua Mukhopadhyay and today I'll discuss about microbiology of protein fibrils and inhibition of protein aggregation through polymer surfactant bioconjugation. So this work has been done in collaboration with Professor Erika Iser from University of Cambridge. So you all know that proteins are most functional building blocks of our body, and because of its unique structural and functional features, they have been used to form different multifunctional responsive materials. Now, but the major challenge of working with proteins are that they are prone to aggregation, and misfolding of proteins leads to aggregation and formation of amyloid fibrils, which are known to be the major causes of different neurodegenerative disorders such as Parkinson's disease and Alzheimer's disease. And typically, this amyloid fibrils formed because of hydrophobic interactions, and they are insoluble. Now, the mechanism of amyloid fibril formation has not been fully understood by the researchers. And because of this, the major focus of the researchers are based on prevention of protein aggregation. Now, prevention of protein aggregation is very important because of its implications in preventive medications and also in therapeutic proteins. For example, therapeutic proteins are known to be the next generation candidate of pharmaceutical industry, and stabilization of the proteins against aggregation is extremely important in the therapeutic protein industry. And also, prevention of protein aggregation is important to functionalize different, fabricate different protein-based materials, for example, protein-based hydrogels and films. Researchers have shown different protein aggregation prevention approaches. For example, side chain chemical modifications, protein sequence modifications, and also small molecule attachment to proteins such as small drug molecules, organic molecules. And also researchers have shown covalent attachment of different polymeric macromolecules to proteins to prevent protein aggregation, such as people have used polyols or polyelectrolytes. But if we look at the current scenario, there are hundreds of drugs and antibodies present which have shown protein aggregation prevention. However, till today, none have shown efficient efficacy in clinical trials. Now, this has been happening from last 25 years. And this is very sad that till the Parkinson's patients have used the same medications what their grandparents used, for example, in 1960s. Now, why this is the case? Because these neurodegenerative diseases are a complex disease, and understanding their mechanism inside the cellular environment is extremely difficult and is not understood properly. And also, there has been no suitable approach present for protein aggregation prevention, which can be called a generic approach to utilize in different proteins. Now, we ask the question, can we provide a generic strategy to prevent protein aggregation? Now, let me first introduce to our approach. So we bioconjugate polymers, a PEG-based polymer surfactant to proteins. So if we look at our bioconjugation approach, we use a two-step polymer surfactant bioconjugation, where in the first step, we perform EDC-mediated diamine coupling to the acidic surface residues of protein to get the positively charged protein. And in the next step, we add an anionic polymer surfactant to form protein polymer surfactant bioconjugates in solution. And it's important to note that our polymer surfactant bioconjugates are stoichiometric bioconjugates. So it's not a blend of polymer and protein. Now, what is the structural behavior of these bioconjugates versus unmodified proteins under aggregation condition in dilute aqueous solution? So we take a native globular protein BSA, and the blue schematic representation is for the native BSA monomers. So when we heat these native BSA monomers under literature known protein aggregation condition for 2 hours, we have seen that after 2 hours, there is almost 4-fold increase in the fluorescence intensity. So this clearly signifies that native BSA after 2 hours forms fibrils in solution. However, when under the same condition, the polymer surfactant conjugates have shown no increase in thiofluorine intensity after 2 hours, and even after 5 days, we have found there is almost no change in the fluorescence intensity. So this clearly states that no fibrillary structures are present in the solution, polymer surfactant bioconjugated solution. We also understood the nature of this polymer protein fibrillation through TEM study, and we found that in native PSA fibrils, there is 50 nanometer fibrillar structures are present after 2 hours of the reaction condition. However, the polymer surfactant bioconjugates, if you look at after 5 days, there is almost no fibrillar type of structure present in the sample. To understand the secondary structural information of the proteins during the aggregation condition, we performed detailed FTIR study and further deconvoluted the data. And we found that after 24 hours under the aggregation condition, native BSA shows decrease of helicity character, and there is significant increase in beta sheet content. So the fibrillar structures are beta sheet nature, we can say. And the polymer surfactant conjugated BSA retains its alpha helical structure after 24 hours. And if we look at the FTIR spectra, even after 5 days, it retains its 55 to 65% alpha helicity character. Just to confirm our thiofluency data and also TM data, we have also performed detailed dynamic light scattering experiments. So if we look at the left panel, our dynamic light scattering experiments with the native BSA solution, so we found that there is an increase in hydrodynamic radii from 4.5 nanometer in native BSA fresh solution to 18 to 20 nanometer in native BSA aggregated state. Whereas if you look at the polymer surfactant conjugated BSA, there is no change in size after 2 hours, and even after 5 days, we have seen that there is no change in the size of the aggregate of the sample, protein sample. And we have further also performed translational diffusion coefficient analysis of the protein samples at 3 different concentrations. And we found that the rate of fibrillation in case of native BSA increases with concentration, whereas the polymer surfactant conjugated part have shown even after 5 days no change in the diffusion coefficients. So this again states that the polymer surfactant conjugated BSA does not show any fibrillation, and the polymer surfactant layer around the protein basically prevents it from aggregation. Now, to understand further the native BSA fibrillation and the mechanical properties of the native BSA fibrils, we have performed dynamic light scattering based microreology to understand the viscoelastic properties. Now, before going in detail of our microreology data, I would like to discuss about the difference between microreology and bulk rheology technique. So bulk rheology technique is essentially a bulk viscosity measurement technique where we can measure strong gels, and it is limited to higher sample volume, and it does not allow local measurements to be performed. But in microreology, we disperse a submicron size colloidal particles inside our sample, and by looking at analyzing their Brownian motion, we can probe their material response on microliter length scale samples with microliter sample volumes of weak gels. So basically, this provides both local measurements and also bulk property measurements. So we perform a dynamic light scattering based passive microreology, where from the instrument, we disperse the 230 nanometer colloidal particles, and we get the intensity from the instrument, we get the intensity fluctuations data, and then we convert the intensity correlation data to mean square displacement. And further, mean square displacement data on applying generalized Stokes-Einstein relation provides both loss and storage moduli. Now, to understand the nature of the native BSA aggregates, we have performed dynamic light scattering based microreology study with native BSA aggregates at 3 different concentrations with increasing time. And we found that both 100 micromolar and 150 micromolar sample with time almost shows no significant change in the MST. value with time. So if you look at the MSD data on the right side, so with time, it's linearly increasing, just as in the case of the response of the particles in proper Newtonian solution. But if you look at the 200 micromolar data, as the time domain increases, so after 6 days, we can see there is a subdiffusive nature at higher time scale in the MSD value. So this clearly shows that as the concentration of native base solution was increased from 100 micromolar to 200 micromolar, the rate of change in viscosity of the medium is also increasing. So we further converted this MSD data to elastic and viscous moduli, and we found, so if you look at the left panels in data, so the blue curve basically indicates the response of the proparticles in water. The black curves represents the response from fresh solutions, and the red curves represents the response from 6 days under aggregated condition of native PSA. So you can see that both G prime and G double prime value increases linearly in case of water and in case of fresh solutions. So this signifies they behave like a Newtonian fluid. Whereas if you look at after 6 days data, G prime value clearly shows a plateau region at higher omega. So this shows that after 6 days, the sample, the NBSA fibrils behaves like a viscoelastic material, and we can essentially calculate its G prime and G double prime parameters using dynamic light scattering based microreology. So this is the first time we show that different viscoelastic properties of such weak gels can be measured using dynamic light scattering based microreology. So we found that the plateau value is around 3.58 Pascal. And when we calculate the entanglement length, we found that it is around 105 nanometer. So native BSA 200 micromolar sample after 6 days under aggregation condition behaves like a semi-flexible polymer. Just to summarize our results, so we highlight a pathway to prevent protein aggregation utilizing polymer surfactant coating on protein surface. Basically, the polymer surfactant layer is preventing two monomers coming into contact and further amyloid fiber formation. And also, our dynamic light scattering based microreology data revealed that native BSA fibrils at higher, longer time scales behaves like a semi-flexible polymer. And in future, we are focusing on studying the rotational diffusion coefficients of these rod-like protein fibers. Finally, I'd like to acknowledge Professor Kamendra Sharma and Professor Eric Iser for their support and guidance. And thanks to my colleagues Ilya and David for working with me in this project. And also thanks to IIT Bombay and Commonwealth Scholarship for providing me funding. And thanks to all of you. And I'd be happy to answer any questions.